Again, thank you so much for the invitation. It's always great to come and then meet with you guys at least the night before to get to know more problems that happen since harvest here. And again, thank you for showing up here this morning. And like I said, there are some issues with like imports and marketing, but I think our job is to help you get the best crop out of the ground, right? Then we deal with other problems later to do it, right? So I started in Crookston in 2014, so had several field seasons now. I used to do 2 applications for managing Sargasso leaf spot, 2014. Now we moved to 5, right? From 2 to 5. I think you guys were doing probably 4 applications, 4 to 5 back in 14, 15. But now I think last season is really, I think, pushing the limit, right? 8 to 9, right? So it's when we talk to our colleagues in Germany, France, Sweden, Norway, Hungary, it's everywhere. I think everybody's saying like, what's happening? Like, I mean, what do we do with that? I think the only thing is just keep doing, I think, what you're doing. You need a little bit of break from the mother nature. That's all we need, like 21 and 23. So, but today I'm not going to talk about Sargasso. I want to spend more time on managing Rhizoctonia, which we know it's a bigger problem for all of us. And then, depending on the time, we will look at some minor diseases. All right. So disease diagnosis is really critical, right? Every year we talk about, especially for the soil-borne diseases, these are almost like lookalikes, right? I mean, they look very similar. You have to have a lot of training for you to identify, right? So these are the 2 common diseases that we have. One is Rhizoctonia, the other one is called Aphanomyces. We do have both in this area here. And look at some of the other samples, right? So these, I say that even for me, it's tricky, because there are no clear signs for both of these diseases here. It's a combination of both. When I get 5 roots from the field, I get like 2 have rhizoc on them, 2 have AF. It's the same story for other fields. But we never find this 2 diseases on the same root, either one or the other, right? So again, diagnosis is really key. Please talk to your agriculturist and send in the samples, right? So sometimes, I mean, they know it's straight shot, okay, it's Rhizoctonia, but if you guys are not sure, it's better get to the lab and then we can let you know what's happening there. Looking at 2025 samples, right? I'm going to talk about only the root samples. Predominantly, more than 50% of the samples were positive for Rhizoctonia again this year, some Aphanomyces, and most of the Fusarium samples came from the northern part of the valley there for the American crystal, right? So again, we'll be able to help in 2026. Again, send in the samples, just get to the bottom of what's happening in your fields, right? So a few things about Rhizoctonia we talked about in the past, but I think the most important thing since 2014, we have several seed treatments that are labeled right now for Rhizoctonia. So I think what we are dealing with later in the season is basically AG group 2-2. There are 2 subgroups there, 2-3B and then 4. If you look at Rhizoctonia in the northern part of the valley where there's a lot of wheat in the rotation, it's mostly 2-4. But you guys with corn and then beans in the rotation or more corn in the rotation, now it's mostly 2-3B. You still have 2-4s, but predominantly it's 3B. But when it comes to how well they can kill the sugar beets, there is really no difference. Both of them can infect when the conditions are favorable, and when it's severe, you lose those beets. The other thing to keep in mind is, I think there are certain fields that you can actually see really big patches of Rhizoctonia that's happening, right? It takes time to build the inoculum in a particular field, right? This year, you got a little bit of rhizoc, a little susceptible variety, you're going to have a bigger spot. And next time around, again, if you didn't realize, again, little weaker variety, the spot gets a little bigger, right? So that's how the soil-borne diseases, they increase in a field. But where does most of the Rhizoctonia stay in the soil, right? So we looked at 60 different fields, across the 3 co-ops. Most of the Rhizoctonia is actually present in the top 4 inches. And when those fields, when you hit those severe, most of the beets are dying later part of the season, that's where you get rhizoc all the way up to 6 inches, right? So I call these like severe risk fields. Now this is where I think we have to use some fungicides. And cultivation, I think for the most part, you're doing a good job, but to get those resistant weeds, you have to do the cultivation, but when you get some soil into the crowns, that's when you get the crown rot, and then it can also cause the root rot there, right? And the tricky thing with Rhizoctonia, again, it can survive as a dormant structures for 2 to 3 years in the soil. So that's how, if you have a susceptible crop in the rotation, beans, corn, it'll keep going, right? So typically what we see with Rhizoctonia is actually Oh, excuse me. This is the pointer here. Just a little necrosis at the soil line. Pretty soon you lose these plants. Again, the young plants are more susceptible compared to the older ones, typically, right? But as you go later into the season, if the conditions are favorable, right, it needs moisture, it needs heat. So you see the same problem. The crowns collapse. You see significant root rot. But again, the petioles can get really black. That's the crown rot infection. At the end of the season, you see these spiders that's lying flat, right? So the classic symptom of rhizoctonia, when you see severe infection on a root, we call this a ladder, right? So if you can see, there is one lesion here, one here. It's a ladder-like pattern there. But what we are seeing is more of the root rot at the bottom, right? The crowns look really clean, especially if you're not doing cultivation, right? That's still rhizoc. Like I said, in the severe fields, it's present from 4 to 6 inch deep, right? So that's where it hits that, and it will start infecting those beets. And at the end of the season, when these beets die, it just makes the same structures as it does on the Petri plates, and it can survive for 2 to 3 years. We call this sclerotia. So that's why you have to take an integrated approach, right, in the rotation crops. So early planting, when the soil is cooler, it doesn't really like it, so it doesn't cause much infection. So early planting is always best to get the best potential, right? So crop rotation, talked about a little bit. Having small grains in the rotation really helps, but I know it's really tricky here. But if we have those susceptible crops, you have to be a little aggressive in managing this disease. Soybeans, right? Very similar story. It can kill that before they emerge out of the ground. And with rhizoctonia, I think the key is it's never just one plant dying. Maybe it's very initial phase, but once the infection keeps going, you see more than one plant dying, right? So you see a row of plants dying here. This was actually on a sugar beet rhizoctonia trial. We followed with soybeans. It's doing exactly the same thing. Navy beans. Again, these plants are really susceptible earlier in the season. Again, the younger, the more susceptible. You see more than one plant dying in a row. That's very critical for rhizoctonia there. I don't have a picture for corn. I think that the problem with corn is you don't see those plants dying. You know, corn, all those roots, they get the infection, but still I think they can tolerate that. You're not going to lose yield in the corn. But really, you know, when you harvest and then when you have all this sclerotia that are formed at the end of the season, they're being carried over to other crops, and you see those hit in the sugar beets there. So the options that we have for managing rhizoctonia in the sugar beet, they're also available in soybeans and edible beans and other crops. So you can consult some of the guides that you picked up this morning, or if you have more questions, you can call my colleague Dr. Dean Malwick with corn and soybean issues in rhizoctonia. He'll be happy to help. So resistant varieties, right? So I think looking at the specialty varieties for rhizoctonia, you have at least 3 varieties that are available for you. The problem with the specialty varieties, they are still susceptible until the beets get to 6 to 8 leaf stage, right? So that's why you still need to have seed treatment, and then on those severe risk fields, we have to do something else. I'm just going to talk about this in a bit here. So at least the 3 different varieties that hopefully you can choose for some fields for 2026 for your growing area. And then when we look at do they really work, the specialty varieties? They do. So if you have a specialty variety here, the blue line, actually for every point increase in disease by harvest, it was losing about 2 tons per acre. And look at the weak variety, the susceptible variety was losing around 3.7, right? I mean, it's very severe infection level that we artificially created this. This is where you have those severe fields need to have the specialty variety. And what can we do at the time of planting, right? We talked about seed treatments and then the in-furrow fungicides. So since 2014, we have 4 different seed treatments that are labeled for managing rhizoctonia. They all belong to SDHI. They inhibit one key enzyme involved in respiration. They do work really well, right? So most of us probably have like Cabina, Vibrance, and Zelterra for like 2026 season. So we evaluated all these 4 seed treatments at the current labeled rates in a field trial in 2025. And then the in-furrow fungicides showed some data in the previous years. But I think one thing to keep in mind with in-furrow fungicides, if you're using a starter fertilizer, make sure that it's compatible so it's not clogging any nozzles, right? So typically I mix my starter fertilizer in 3 gallons of water and then mix fungicide with 3 gallons of water and then apply 6 gallons down the drip tube into the furrow. But in 2025, we have not used any starter fertilizer because that ground already has really good nutrient status there. So we looked at the several QOIs, SDHIs, and other combinations, right? Quadrus really works. Asteroid, it's another formulation that mixes well with the starter fertilizer. And then Elatus, it's a combination of QOI and SDHI, Headline, Proline, Propulse, and Preaxle, right? So we looked at all of these options in 25. And then we're getting more and more questions about, biologicals, like, okay, do this really work? So this is only year 2. We included most of the bacillus-based biologicals, Ziranar, Bexporn, and Serenade. And then Howler, this is based on, Pseudomonas, right? So looking at the data for this 25 trial, so I'm just showing the stand counts at the earlier part of the season. So this is days after planting on this axis and the number, right? So ideal stands are about 200 plants per 100 foot of row. Look at that. We inoculated this trial and then there was no Rhizoctonia seed treatment. Still we had like 200 plants per 100 foot of row. Just tells that the season was really dry at the earlier part of the year, so we did not see much infection in this particular trial. But what we observed was, some differences. So when I see this as a group, if you look at that yellow line, all of the in-furrow biologicals, they're really safe, during the emergence stage. And then by the time we go to the 5 weeks, they're slightly lower than the seed treatments, which is that green line there. But even in the absence of starter fertilizer, we saw a little bit of reduction in emergence compared to biologicals, but the trend was really maintained by 5 weeks after planting, right? Still excellent stands for both of these in this particular trial. And then at the end of the season, we rate about 20 roots from each plot using a 0 to 10 scale. 0 is a healthy root, and then 10 is completely dead one, just to guess how much severity that we have in those plots. So non-treated control, about 9% severity, but still we are scoring mostly ones, not much happening by the end of the season. 8,500 pounds RSA right here. The seed treatments, and then in-furrow fungicides, they were slightly lower in terms of severity compared to biologicals, and the sucrose was slightly higher for the seed treatments, but really no big differences for the root yield and extractable sucrose. Like I said, the disease pressure was not as high in 25, so most of them looked okay. And the other option we have is post-emergence fungicides, right? So before we had seed treatments for Rhizoctonia, the recommendation was when the 4-inch soil temperature hits 65 degrees, we have to do your post-emergence application. But with the seed treatments, now we say that the best time to apply these fungicides is between 4 and 8 leaf stage, right? And the other thing that changed, right? So most of us are doing like no band sprayers, still doing broadcast applications. So we're doing this trial for a few years now. Again, we conducted in 25. Some of the options that we have for post-emergence control of Rhizoctonia, we looked at both 7-inch band and broadcast application. As you can see, all the fungicides except Excalia, this is the only one you have to use different rates. If you're using 7-inch band and a 22-inch rows, you have to use a third of rate, 0.64 fluid ounce. If it's full rate, it's 2 fluid ounce for broadcast. And 4 and 8 leaf stage, right? When I say 4, if you have like high risk field, a little bit weaker variety, I think go with the 4 leaf stage. But if you have low to moderate risk, you can go with the 8 leaf stage application for these fungicides. Again, looking at the data for this particular trial, so I have my non-treated control. There's more disease here because we actually inoculated these with Rhizoctonia in the middle of the June when we applied this fungicide. So that's why you see a little bit more disease. About 25 tons, and 7,500 pounds RSA for this one. And look at both band and broadcast. So I had about 50% of the roots that are infected with Rhizoc compared to 11 and 8%, 27 to 28 tons, right? So you get about 520 to 900 pounds of ESA or non-treated control with the post-emergence fungicides. So both methods are working well. Again, if you still want to do your broadcast application, just go ahead and do it. Because the part of the reason we started doing this, if you don't have a band sprayer, I don't want you to not to spray for Rhizoctonia. Then you're going to lose more doing that way. I'm really sorry. It looks like my slide format has really changed here. So the font is moving all around here. But again, all the options that we have for seed treatments, in-furrow, and then the post-emergence by the mode of action, different color, right? So just like any other disease management, using different modes of action at different times is really key, right? So you can go with SDHI for the seed treatment, and then maybe come back with the QOI, then use an SDHI as a post-emergence application. That's a good disease management practice, especially in terms of fungicide resistance. Just to summarize, we talked about seed treatments, in-furrow fungicide, and then the post-emergence fungicides. These work really well, but again, the key is we do it in 4 or 8 leaf stage depending on the severity in the field. So for the most fields, I would say, you order your seed, it comes with the seed treatment, then you can do a post-application, but otherwise you can add the in-furrow at the time of planting for those severe risk fields. I want to switch gears and talk about Aphanomyces a little bit, right? So I showed this picture earlier. So the key thing with Aphanomyces, it does not kill the beets before they emerge out of the ground. It's only after, post-emergence. You see pinching at the soil line, pretty soon you lose those seedlings to the really, high winds that you have. They will end up in the neighbor's field. That's how you know you have Aphanomyces. And another thing, we don't see the ladder-like pattern for Aphanomyces. It really tracks the moisture. You see this root rot, really large necrotic lesions on the roots here. Again, if you have infection earlier in the season, and then if the rest of the season stays dry, you don't see any of the active infection on those roots. But what you see is the malformed roots. It means like you're missing little pieces of those infected roots from earlier part of the season there. So same thing. It does not really cool soil temperature, so early planting really helps. And then we have 2 excellent seed treatments for managing Aphanomyces. They work very well, as well as the specialty varieties, right? They do work for Aphanomyces as well. So just look at the picture here. This is from the beta seed Aphanomyces nursery in Shakopee, right? So really stark difference between the resistant variety, which is a specialty one, and susceptible variety. There's hardly anything left by 3rd week of August in there. The other thing we can do for Aphanomyces is actually applying the factory waste lime. I know most of you are putting about 5 to 6 tons every 3 to 4 years, but as you can see, with every ton of lime that we apply, we can really drive the disease down by the end of the season here. Again, this trial was done in Breckenridge. We can actually see the benefit of lime, just a single application, even working after 12 years. So nothing lasts for that long, right? So again, wish I had checked the slides how they were coming here, but there are a few diseases that we picked up in the last few years. Again, these are not major concern, but since we saw positive results from our disease diagnosis, we want to make sure that we're able to control these with what we are doing with Cercospora leaf spot, right? So typically, if you have a field with stressed beets, you can really get Alternaria leaf spot on that. As you can see here, little round ones are the Cercospora ones, and these are all caused by Alternaria. And on top of Alternaria leaf spot, we were also seeing another pathogen. It's called Stemphyllium leaf spot. So you can see actually in the same spot, Alternaria is growing here, Stemphyllium is growing here. So we did a field trial in 24 and 25 in Crookston to really look at, is there any difference in terms of the varieties? Let's say if you have a susceptible variety for Cercospora versus CR plus varieties, because the timeline we saw these diseases were really in alignment with when the CR plus varieties came. So we used 3 different varieties, and then we artificially created the disease pressure in this particular trial. And the number 2, when we applied fungicides to manage Cercospora in this, do they also manage Alternaria and Stemphyllium? So I'm going to show something from 2024 here. So even though we did not inoculate Cercospora in this particular trial, it came in naturally by middle of July and established in the canopy. As you can see, when we look at the overall leaf spot in this trial, the green line is actually, that's a total leaf spot for the susceptible one, and then the purple one is, and the red one are the 2 CR plus varieties, right? So when I break it down, what is this that we are seeing in terms of the leaf spot? Again, the green one is actually the Cercospora leaf spot, and the red one is Alternaria, and then the purple one is the Stemphyllium, right? So predominantly it's Cercospora, but there's more Cercospora in the susceptible variety compared to CR plus. This is in 2024. And looking at the impact of the standard 4 spray program, so this trial was planted a little bit later and inoculated later in the season, so only 4 sprays were applied here. The standard program using DMIs and then contact fungicides. As you can see, again, there is more disease in the non-treated control where fungicides were not applied compared to standard 4-spray program. And again, if I break it down, what are we seeing in those two non-treated and the standard spray programs? Most of it is the Cercospora leaf spot, a little bit of alternaria here, and then stem flea is very little in this. So same trend was observed for 2025 as well. So really, I think as long as we're doing a good job of managing Cercospora leaf spot, we can also take care of alternaria and stem phyllium. That's an indirect benefit that you get. So with that, I think that's kind of the same thing I just talked about in the 2 slides there. I would like to acknowledge the R&D board for funding these trials, and then the NWROC core team, the co-ops, and the Germancy technology, crystal sugar, everybody. And then last but not the least, the hardworking team from the Sugarbeet pathology team in Crookston. So with that, thank you. I don't know, Mark, if I have time for a question.
Yep.